Chapter 2, Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In lineage time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that include all plant and animal respectively. This system did not distinguish between the eukaryote and prokaryote, unicellular and multicellular organism, and photosynthetic green algae and non-photosynthetic fungi organism. Classification of organism into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into other category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over the time. Through plant and animal kingdom have been a constant under all different systems. The understanding of what group organism be include this kingdom have been changing. The number and nature of other organisms have also seen by understood differently by different scientists over the time. Table 2.1, Characteristics of the five kingdom. Five kingdom characters: Monera, Protista, Fungi, Plantae, and Amelia. Cell type: prokaryotic, eukaryotic, 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 eukaryotic. Cell wall: non-cellulosic, polysaccharide plus amino acid present in some, present with chitin, present cellulose, absent. So nuclear membrane: absent, present, 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 present. Body organization: cellular, cellular, multicellular, loose tissue, tissue, organ system. Mode of nutrition: autotrophic, chemosynthetic, and photosynthetic, and heterotrophic, saprophytic, and parasitic. Autotrophic, photosynthetic, and heterotrophic, saprophytic, parasitic, autotrophic, photosynthetic, heterotrophic, holozoic, saprophytic, etc. R.H. Whitcomb, 1969, proposed a five-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of the five kingdoms. The three-domain system have also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryote kingdoms in the three-domain and thereby a six-kingdom classification. You will learn about this system in detail at a higher classes. Let us look at this five-kingdom classification to understand the issues and consideration that influence the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, fern, gymnosperm, and the angiosperm under plants. The character that unified the whole kingdom was that all the organisms included had a cell wall in their cell. This placed together group which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other group were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say for example, Chlamydomonas and Spirochara were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plant, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plant had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista have brought together Chlamydomonas, Chlorella, earlier placed in algae within plant, and both having cell walls, with Paramecium and Amoeba, which were earlier placed in the animal kingdom, which lacked cell wall. It was put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationship over time. An attempt has been made to evolve a classification system which reflect not only the morphological, psychological, and reproductive similarities, but is also phylogenetic. Example, it is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whitcomb system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, we will deal separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow, and deep ocean, where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, plural cocci; the rod-shaped bacillus, plural bacilli; the comma-shaped vibram, plural vibro; and the spiron-shaped spilum, spila, plural spirilla. Figure 2.1. Cocci, bacilla, spirilla, vibro. Bacteria through the bacteria structure is very simple. They are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. For example, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotroph. For example, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot spring, thermoacidophiles, and marshy area methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methogens are present in the gut of several ruminant animals such as cow and buffaloes, and they are responsible for the production of methane biogas from the dung of this animal. EU bacteria. There are thousands of different EU bacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The kind of bacteria, also referred to as blue-green algae, have chlorophyll A similar to green plant and are photosynthetic autotroph. Figure 2.2. The kind of bacteria are unicellular, colonial, or filamentous freshwater, marine, or terrestrial algae. The colonies are generally surrounded by glantaceous sheath. They often form blooms in polluted water body. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyte. Example: Nostoc and Anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrite, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotic, fixing nitrogen in ligand roots. Figure 2.2. A filamentous blue-green algae, nostoc. Figure 2.3. A dividing bacterium, roots, etc. Some are pathogen causing damage to human beings, crops, farm animals, and pets. Chlora, typhoid, tetanus, citrus canker are well known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3. Sometimes, under unfavorable conditions, they produce spore. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animal and plants. Kingdom Protista. All single-cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistian to one biologist may be a plant to another. In this book, we include chrysophyte, dinoflagellate, euglenoid, slime module, and protozoan under Protista. Members of Protista are primarily aquatic. This kingdom form a link with the other, dealing with plant, animal, and fungi. Being eukaryotic, the protistian cell body contain a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes. This group include diatoms and golden algae desmide. They are found in fresh water as well as in marine environment. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatoms, the cell wall from two thin overlapping shell which fit together as in a soap box. The wall are embedded with silica and thus the wall are indestructible. Thus diatoms have left behind large amount of cell wall deposit in their habitat. The accumulation over billion of years is referred to as diatomaceous earth. Being gritty in this soil is used in polishing, filtration of oil and syrups. Diatoms are the chief producer in the ocean. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cell. The cell wall has a stiff cellulose plate on the outer surface. Most of them have two flagella. One lie longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellate, example gonologs, undergo such rapid multiplication that they make the sea appear red. Red tide toxin released by such a large number may even kill other marine mother animals such as fishes. Euglenoids, majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoids are identical to those present in higher plant. Example: Euglena. Figure 2.4b. Slime module. Slime module are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material under suitable condition. They form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable condition, the plasmodium differentiate and form fruiting bodies bearing spore at their tip. The spore possess two walls. They are extremely resistant and survive for many years, even under adverse conditions. The spore are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoan. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet, as in amoeba. Marine forms have silica shell on their surface. Some of them, such as entamoeba, are parasite. Flagellate protozoan. The members of this group are either free-living or parasitic. They have flagella. The parasitic form causes diseases such as sleeping sickness. Example: trypanosoma. Ciliated protozoan. These are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity gull that open to the outside of the cell surface. The coordinate movement of row of cilia causes the water laden with food to be strayed into the gut. Example: paramecium. Figure 2.4d. Sporozoans. These include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruit. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, for example yeast, are used to make bread and beer. Other fungi cause disease in plant and animal. Wheat rust causing peucania is an important example. Some are the source of antibiotics. Penicillium fungi are cosmopolitan and occur in air, water, soil, and on animal and plant. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is prevent even to food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular fungi, are filamentous. Their body consists of long cylinder thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called conocytic hyphae. Others have separate or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence are called saprophyte. Those that depend on living plant and animal are called parasites. They can also live as symbionts in association with algae as lichen and with root of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidae or sporangiospore or zoospores, and sexual reproduction is by oospore, ascospore, and basidospore. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second, fusion of two nuclei called karyogamy. Third, meiosis and zygote resulting in haploid spore. When a fungus reproduces sexually, two haploid hyphae of commutable mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2N. However, in other fungi, ascomycete and basidomocyte, an intervening dikaryotic stage and plus an example, two nuclei per cell occur. Such a condition is called the dikaryon and the phase is called dikaryophase of fungus. Later the parental nuclei fuse and the cell become diploid. The fungi from fruiting body in which reduction division occur, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. Phycomycetes. Members of icomycete are found in aquatic habitat and on decaying wood in moist and damp places or as obligate parasite on plant. The mycelium is aseptate and conocytic. Asexual reproduction takes place by zoospore motile or by alpanospore non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similarly in morphology isogamous or dissimilar anisogamous or oogamous. Some common examples are meteor figure 2.5a, rhizopus, the bread module mentioned earlier, and albago, the parasitic fungi on mustard. Ascomycete, commonly known as sec fungi, the ascomycete are mostly multicellular. Example penicillium or rarely unicellular. Example yeast, Saccharomyces. These are saprophytic, decomposer, parasitic or coprophilous, growing on dung. Mycelium is branched and separate. The asexual spore are conidae produced exogenously on the special mycelium called conidae spores. Conidae on germination produce mycelium. Sexual spore are called ascospores, which are produced endogenously insect like asci, single ascus. These asci are arranged in different type of fruiting bodies called ascocarps. Some example are Aspergillus figure 2.5b, calvicaps and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morel and truffle are edible and are considered delicious. Basidomocytes, commonly known form of basidomocyte are mushroom, bracket fungi, or puffball. They grow in soil on logs and tree stumps and in living plant body are parasites. Example rust and smurfs. The mycelium is branched and separate. The asexual spore are generally not found, but vegetative reproduction by fragmentation is common. The sex organ are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cell of different strain or genotype. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, produce four basidospore. The basidospore are exogenously produced on the basidium, plural basidae. The basidae are arranged in fruiting body called basidocarp. Some common members on are agricus mushroom figure 2.5c, Ustiglosmut and Pucania rust fungus, deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phase of this fungi are known. When the sexual form of this fungi were discovered, by they were moved into classes they rightly belong to. It was also possible that the asexual and vegetative stage have been given one name and placed under deuteromycete and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of deuteromycete. Once perfect sexual stages of member of deuteromycete were discovered, they were often moved to ascomycete and basidomocyte. The deuteromycete reproduce only by asexual spore known as conidae. The mycelium is separate and branched. Some members are saprophyte or parasite, while a large number of them are decomposer of litter and help in mineral cycling. Some examples are Alternaria, Colymbrichium, and Trichoderma. Kingdom planting. Kingdom plant include all eukaryotic chlorophyll containing organism commonly called plant. A few members are partially heterotrophic such as the insectivorous plant or parasite. Bladderwort and Venus flytrap are example of insectivorous plant and custata is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plant include algae, bryophyte, pteridophyte, gymnosperm and angiosperm. Life cycle of plant have two distinct phases, the diploid sporophytic and the haploid gametophytic that alternate with each other. The length of the haploid and diploid phase and whether these phases are free, living or dependent on other vary among different group in plant. This phenomenon is called alternation of generation. You will study further detail of this kingdom in chapter 3, Kingdom Animalia. This kingdom is characterized by heterotrophs, eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly dependent on plant for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher form show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent features of various phyla are described in chapter 4. Virus, viroid, and prions and lichens. In the five kingdom classification with care, there is no mention of lichens and some acellular organisms like virus, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effect virus can have on us even if we do not associate it with our condition. Virus did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having a inert crystalline structure outside the living Figure 2.6 Tobacco mosaic virus TMV bacteriophage. Once they infect a cell, they took over the machinery of the host cell to replicate themselves, killing the host. Would you call virus living or non-living? Virus means venom or poisonous fluid. Dimitri Levinsky, 1892, recognized skeleton microbe as causal organism of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they pass through bacteria-proof filter. M.W. Bergeric, 1898, demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogen virus and called the fluid as Contagium vivium fluidium, infectious living fluid. W.M. Stanley, 1935, showed that virus could be crystallized and crystal consists large of protein. There are inert outside their specific host cell. Virus are obligate parasite. In addition to protein, virus also contain genetic material that could be either RNA or DNA. No virus contain both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious in general. Virus that infect plant have single stranded RNA and virus that infect plant either have single or double stranded RNA or double stranded DNA. Bacterial virus or bacteriophages, virus that infect the bacteria are usually double stranded DNA virus. Figure 2.6b. The protein coat called capsid made of small subunit called capsomer protects the nucleic acid. This capsomer are arranged in helical or polyhedral geometric form. Virus causes diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing, and vein clearing, drafting, and stunted growth. Broids. In 1971, Theo Dunier discovered a new infectious agent that was smaller than virus and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name broid. The RNA of the broid was of low molecular weight. Prawns. In modern medicine, carried infectious neurological disease were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prawn. The most notable diseases caused by prawns are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant CR Jacob disease (CGD) in humans. Lichens. Lichens are symbiotic association. Example: mutually useful association between algae and fungi. The algal component is known as phycombioint and fungal component is mycobioint, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if we once saw lichen in nature, one would never imagine that they have two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas. Summary. Biological classification of plant and animal was first proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whittier proposed an elaborate five-kingdom classification, Monera Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition, and reproduction, and phylogenetic relationship. In the five-kingdom classification, bacteria are included in the kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-cell eukaryotes such as plasmophyte, dinoflagellate, eugenoid, slime module, and protozoan. Protists have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of kingdom Fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycete, ascomycete, basidiomycete, and eutromocyte are the four classes under this kingdom. The plant include all prokaryotic chlorophyll-containing organisms. Algae, bryophyte, pteridophyte, gymnosperm, and angiosperm are included in this group. The life cycle of plant exhibit alternation of generation, gametophytic and sporophytic generation. The heterotrophic eukaryotic multicellular organism like a cell wall are included in the kingdom Animalia. The mode of nutrition of this organism is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like virus and viroid as well as the lichen are not included in the five kingdom system of classification.